Hello everyone, I am Gaurik Mukherjee. Today, I'll be presenting the work that I am doing with Professor Leelavati Narlikar on leveraging single-cell data to simultaneously identify and characterize regulatory heterogeneity based on motifs in mixed cell populations. This research is made possible through the support of research institutions like IISER Pune and CSIR National Chemical Laboratory in India, and as part of the National Network Project funded by the Department of Biotechnology of the Government of India. In the context of developmental and cancer biology, researchers frequently encounter cells that are highly heterogeneous in nature. Although the genetic code remains largely conserved across cell types, variations in transcription factor combinations can lead to distinct transcriptional profiles and, consequently, cellular diversity. Here, transcription factors regulate gene expression by binding specifically to recurring nucleotide sequences in the DNA called motifs. Now, since the binding sites of transcription factors typically reside within accessible regions of chromatin, we decided to use single-cell ATAC-seq. Single-cell ATAC-seq provides a genome-wide chromatin accessibility profile of the cell. This pattern variability in chromatin accessibility is popularly used to identify regulatory heterogeneity in cell samples. However, most of the existing methods do not incorporate any information about the underlying motifs. Furthermore, the computational analysis of single-cell ATAC-seq is far more difficult due to the data's high dimensionality and sparsity. Hence, we figured that by using gain or loss of chromatin accessibility within sets of transcription factor binding motif combinations, we can create a more comprehensive and dense regulatory profile of the cell, which can better understand and categorize groups of cells that exhibit similar behaviors within the profile population. To do that, we leveraged sequence information in ATAC-seq peaks and scanned for occurrences of known transcription factor binding motifs. Then we incorporated that motif information with chromatin accessibility profiles in single-cell ATAC-seq while controlling for technical biases in the epigenomic data. We termed this the motif activity profile of the cell, as the cell's regulatory profile now has motif combinations in the feature space. A method called Chrome-VAR uses a similar approach, but it only accounts for single motif occurrences, which may not capture all the necessary regulatory information since transcriptional regulatory programs are typically driven by multiple transcription factors working in concert. After we construct the activity profiles, we can use it to cluster the cells and identify the different cell groups. Since transcription factor binding motif combinations serve as the features in this context, any feature selection method can be directly employed to simultaneously also identify key motif combinations in each cell group. These are some of the tools that we have used for our analysis. We use Jasper for the motif database, FIMO, which is short for Find Individual Motif Occurrences from MemeSuite, was employed for sequence analysis, and R and Python were used as the programming languages for the analysis. To evaluate the performance of our pipeline, we applied it to real-world single-cell ATAC-seq datasets. One representing the epigenomic profiles of differentiating hematopoietic cells in humans, and the other capturing the epigenomic profiles of key developmental stages in Drosophila melanogaster embryogenesis. First, we wanted to check whether a regulatory profile that captures chromatin accessibility changes within combinations of transcription factor binding motifs can actually perform better than the profile that captures these changes only within individual motifs. So we constructed the corresponding motif activity profiles for both these cases and performed supervised machine learning using sparse multinomial logistic regression, or SMLR, to predict cell types given their activity profiles. SMLR also performs feature selection for each cell type, pruning irrelevant features or motif combinations that do not significantly impact cellular identity. We ran the experiment across different training test splits while using accuracy as the scoring metric and constructed box plots for the same. By looking at the box plots, we can clearly see that by having a regulatory profile that captures chromatin accessibility changes within motif combinations has enabled better categorization of the groups of similar cells. SMLR also provides feature weights for each cell type, which indicates how strongly each feature or motif combination is associated with or distinctive for each specific cell type. In Drosophila, we discovered that VFL-TRL motif combination has high feature weight during early embryogenesis. This finding is consistent with literature, as Zelda, which is the protein that VFL codes for, together with GAGA factor protein, which TRL codes for, they both together drive early zygotic genome activation and chromatin accessibility in Drosophila melanogaster embryo. In summary, we showed that using gain or loss of chromatin accessibility within sets of motif combinations can better explain groups of similar cells within the profile population, while also enabling simultaneous identification of key motif combinations within each group. Thank you.